Quantitative Investigations of Variation, AQA Biology. Key terms. Intraspecific variation is when one species differs from one another. Intraspecific variation is when members of the same species differ from one another. The mean is when you add up the total and then divide by the amount tested. The mode is the most common number. Medium is the central value when put in order. And standard deviation measures the spread about the mean. Measuring distribution. This is done using quadrats and transects. A quadrat would be used regarding if it's a suitable size, random sampling is used, and you identify plants by measuring the abundance. And then transects are used by using the tape measure and recording species at regular intervals by placing quadrats down. Now, you might be wondering why I'm not going into too much detail about this, but that's because it's more so A2 content than AS. So, what is the purpose of sampling? To take measurements of individuals selected in a population of organisms, and individuals are a representative of the population being investigated. So what are the disadvantages of sampling? Well, you have sample bias and chance. Sample bias would be things such as everybody's going to want to go and take measurements in the nice green meadow instead of the dark green bush that has stinging nettles and thorns and such. So you actually being biased because there actually might be more variation in that thorn bush than there is in the meadow. And then chance. You could be in an area that is full of bare ground, but you just so happen to pick the coordinate in which has vegetation on it, which isn't a true representation of the total population. So how would we minimize the effect of chance in sampling? Well, we use a bigger sample size and use data analysis techniques. So how would we measure species diversity? Species diversity is measured using the following calculation. So you have D for species diversity equals N bracket N minus 1 divided by, oh, that's not a nice line, the sum of small little n, open bracket, N minus 1. Now you might be thinking, what the actual is this? Well, the D, I'm not going to write them down. Oh, I'll try. Okay. We're going to say is species diversity. So, species diversity. I'm so sorry, but I'm doing this on my laptop, so I can't write it out. This would be, say if you had, okay, counted species. So you have count okay guys, I know it should be the other way around, but I'm really pleased. Okay, so species, let's say you have flowers, I'm going to represent with F, grass, and shrubs. So you have 10 flowers, 8, let's not call this graph, let's call this, what we'll call this graph. A second. And then 9 shrubs. So your big N would be 3. Because there are 3 different species. your small n would be these numbers. So how you would calculate this, you do 10 open brackets times 9, then 8, that was a horrible 8, times 7, and then 9 times 8. Please, I can't do 8s on this. I'm working on my laptop, sorry. When you'd add these together, you'd come out with the total, which is this, which is sum of. So when you'd add these together, you'd get the answer of 218. So what you would do this, you would do 3 bracket 2 close bracket would equal your species diversity, which in this case, you would never get a question this simplified, but would be an answer of 0.028, which is a very small number. You can get, the closer it is to the value, the more positive the value, not more positive, but the bigger the value, the more diversity you have. Threats to biodiversity. Now, I like to use a little acronym to remember this. So, actually, let me go through what it is first. So you have habitat destruction, invading species, population increase, pollution, and overexploitation. Now, can anyone guess what the acronym is? Looking at the picture. Well, yeah, it's called HIPPO. So H, habitat destruction, I, invading species, P, population increase, P, pollution, O, overexploitation. So now there are human threats to biodiversity, such as agriculture, which can alter the landscape and trigger eutrophication, deforestation, and exploitation of resources. But these are the main ones that you probably get marks for in your exam. Now, standard deviation is something you'll probably see in your exam, which are usually in graphs such as this. So you'll have maybe your bar charts, and your standard deviation will be shown like this, using these symbols. Now, this represents the spread of numbers around the mean. And if these standard deviations overlap, they mean that they aren't significant. And the difference between the two values are that they're due to chance. And what you would say, you say this at a probability level of 0.05. 0.05. Now you might be wondering, why is this relevant? Like, if it's 0.05, then you're not that sure, are you? Well, actually you are, because this actually equates to being 95% certain that what you've said isn't due to chance. And it is a significant difference. So if you had a question in your exam and it didn't have standard deviation bars in it or error margins, then one thing you need to do is question why. it doesn't have these doesn't show standard deviation, and question the reliability of your results, and that could be worth a few marks in your exam. So I hope this is useful, and this is actually the end of the AAS units. If you like them, please like and subscribe, and the next video will be about photosynthesis.